Welcome to FlowStars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry, brought to you by Beckman Coulter at Bitesize Bio.
Welcome to FlowStars, and today I'm joined by Sofie Van Gassen from VIB. She shares her motivations for getting involved with flow cytometry. As someone, one of the fastest rising stars in flow cytometry, shares her vision for the future of data science and the quality she thinks a good principal investigator should have. All this coming up in FlowStars. Hi, welcome to this special edition of FlowStars. I'm Peter O'Toole from the University of York, and today I'm joined by Sofie Van Gassen from the VIB in Ghent. Sofie, how are you?
Hi, I'm doing well. Thank you so much for having me today.
That's okay. I don't think we've ever met in person, and yet I know about your work, and it's work that I don't think I could ever replicate or duplicate. So actually, do you want to just very quickly start for the listener or the viewer? What is it? Where's your area of expertise?
So I have a background in computer science, actually. So not in immunology, not in anything biology related. And so I've been working in bioinformatics. Actually, the topic of my PhD was machine learning for flow cytometry. So then when I was considering doing a PhD, I got introduced to this technology. And since then, I've been working on flow cytometry data, and I've been enjoying it a lot. So algorithm development, also developing pipelines for existing data sets, things like that. Yeah.
Did you not say just before we started recording that you're not good at remembering numbers?
Yeah. So that's why the computer remembers them all for me, right?
Okay, fair enough. Like I say, for someone who plays with lots of numbers, that's kind of an interesting problem to have with it. So you did a PhD, and you would give it No, you must, because your PhD must have started with flow cytometry, surely.
Yeah. So the topic of it was really flow cytometry. So basically, Ivan Seist, my professor, just noticed that people were doing all this manual analysis of this data. People he was collaborating with, and was thinking there seems to be room in here for exploring some algorithms, seeing whether we can help these people with automating some of these tasks, things like that. And so he proposed this topic, and then we were immediately collaborating with the people actually measuring the data. I think that's been a huge benefit for me, that it was not just like in a computer science group, a theoretical exercise, like, oh, let's look at this data points and try to find some clusters in there, but that we were actively talking to the people who were generating the data and which questions and problems they were facing. And so that's been the whole focus of my PhD, and actually of my work still. I think I'm one of those few people who kind of keep doing what they're doing after their PhD. Like I'm now already a number of years a postdoc, but I'm still doing kind of the same things as I did during my PhD. And of course now a bit more guidance than at the start of my PhD, of course, when it was mainly the programming. Topic-wise, it's still very closely related.
It's an interesting area to have chosen as a PhD student, to know the importance, relevance, or to be inspired to do flow cytometry. As a computer scientist, the world is your oyster. Why did you think flow cytometry was for you?
Actually, I was very hesitant to do a PhD, and my professor had to kind of convince me, because I wanted to do something applied. I wanted to do something that was used, and I was a bit afraid if you do a PhD in computer science, you're just like reasoning about algorithms, and there's lots of value in that, but it would not be for me if it was just a theoretical exercise. But then he convinced me, like these people that I'm collaborating with who are facing these specific issues, and if you work on this, you could actively be involved in that and help them. And so that applied aspect of it, even though it's still then applied in fundamental research, of course, but the applied aspect of the algorithm side of things is what really convinced me. He convinced me, like, if you work in this, people will use your tools. And actually that turned out to be the case by now. So that's really cool. And that's also really where I get my motivation and my things to do this. Yeah, absolutely.
So I can see just by your smile, your eyes, your enthusiasm that you're loving the work, the environment.
Absolutely.
And quite often So I've employed lots of data scientists over time. And I quite often hear, oh, yes, but you'll never get a good data scientist because they can earn lots more money working for Google or the banks or someone else. I always disagree. But go on, why have you chosen this instead of going earning lots of money in other areas?
I think the combination of the fact that it's just really interesting and really cool to do, like that once you're in there, you want to keep working on it. I think the fact that you know that you're working in this fundamental research, but also health related, that it can have an impact on society. And of course, like it will not be my algorithm in itself, which immediately impacts the whole society, but like you're one small radar in this whole framework, but you're aiming towards that. So the fact that it's life science related, even though I don't have like background in that, I didn't study any biology, immunology, but the fact that you know that you can contribute to that field also gives me a lot of motivation. That in the ultimate dream is, of course, that you help patients, right? That you make people's life better, and give them better opportunities for their health. And I'm very far away from that. I'm not a medical doctor. I'm not someone with a biological knowledge, but I know I can contribute to that, and that also would motivate me way more than, for example, optimizing advertisements or something like that, which indeed you could also do from the computer science point of view.
I'll big it up even more because actually so many of the medical diagnoses are through flow cytometry itself, that actually your analysis, the new analyses that you're enabling on flow cytometry could have a profound impact. And so you say, Jess, you're not a doctor in a clinical doctor sense, but they are utterly dependent. All they do is take a sample and then tell the patient what you've enabled them to say. So I would say that actually,
Well,
you're only a clinical doctor. You're the
I hope there's
scientific
some reasoning
practitioner.
in between somewhere as well, right? No, but that's exactly what I mean, right? You know that your work has an impact and that it can be used by these people who have the tools to then further interpret these things and bring them to the patient. And that's really cool.
Actually, another question then that strikes me. So throughout your PhD, now your postdoc at VIB, how many different groups are you getting data from?
Oh, that's an interesting question. I don't know if I've counted that. So at the beginning, we were mainly collaborating with this this one group that my professor was already collaborating with, but this very quickly actually extended through collaborations, through meeting people who had questions. And I think it depends a bit also on how you define it. So I think there's a number of groups that we're actively collaborating with. I would say that's, oh, yeah, a reasonable number, 3, 4, 5, 6, something like that. But then you also have many people reaching out with a singular question, right? Who have like, oh, I'm trying to use your tool, but we're encountering this or that. Can you give some advice? And that's actually, of course, it takes a smaller amount, it's not a long-term commitment, but that way we get asked about lots of data from lots of different groups. And then once in a while, we're working also on some public data. That also comes with its challenges. I'm a very strong advocate for making data public. I think this will really help the field. But just finding public data online where you don't know much about it and don't have insights about the metadata, about what was going on while measuring things, can also be an extra challenge. So I appreciate it a lot if the people with a biological question are also involved in the project rather than just working on data that is out there.
I think what's quite interesting is you're doing a postdoc, but it's not so dissimilar to a core facility in that the number of people you're helping and advising, and at the top end of a core facility, you're helping design experiments, you're helping analyze the data, helping really make their science fit around the technology. And here you are, the other side of this, downstream, because then they get all this analyses. And actually, from my side, I'm utterly useless when it comes to the coding side. And there you are to pick that up, which is a high-end problems.
Yeah. Actually, that's an interesting question, because that's kind of where I see my future going. Nothing's on paper yet, but you can't keep doing postdocs forever. And I don't have the ambition to go for a professor position at this point. Never say never, but I don't really expect that happening. So I think right now I'm mainly then giving guidance in the algorithm development and so on. I notice that we also get lots of questions of users who want guidance in how to use these tools. And I'm actually talking to our flow core facility whether there could be options there to give more support also from the core facility, and then have a future position there, hopefully, if everything works out. So fingers crossed.
I presume that's with Kurt that's running the core facility at VIB?
Exactly.
I've got two lines of thought now. I don't know which way to take this in first. Firstly, I'd say actually not going down the academic route. So something we've done at York is set up a data science hub. And really, I would argue that they're applied academics. And I think this is really important because when it comes to helping with the data science side, there's always talk about bringing new data science lecturers in, so new group leaders in data science. But naturally, they have their own niche area of data science that they want to develop. And that's not necessarily broadly applicable to the wide user base that would be using the flow core at the VIB, for example. Whereas actually, if you're more applied in the academic world, you can develop new algorithms, new solutions, but you have a whole shop of different shelves to pick problems off and to work. And the problem is you can't pick and mix. You kind of have to be you have to deal with what's put in front of you. But I've got to say, I think it's a vital role that's no less important than a primary academic. And certainly at York, we're trying to make that a peer-to-peer, that it's not academic in that you are writing your own grants necessarily, but you're co-eyeing on grants, you're part of the grant team, you're part of the project team. So very much an applied academic career in a technical setting.
Yeah. No, that's a really cool concept. I would kind of argue that's, well, not argue maybe, but think that if you're a good PI, you also look for these collaboration opportunities and you look for possibilities to work together and to actually see where your field can contribute to the questions that people are having. As a data analyst researcher, I think that's also what I see happening in our group. So it's the case for the flow cytometry research we're doing, but we're also working on some other technologies, like not me personally, but other people in our group with transcriptomics, cell communication, all these things, spatial things now also more and more. And I think that's also part of the strength and the success of our group, is that we're really looking at where are biologists asking questions, where are the boundaries of what's currently possible with the techniques that are out there now, and where can we still contribute. And I think that's really what's as you would hope for, that these other PIs would also aim to do, rather than just staying focused on like, I'm doing this one thing. Of course, you have expertise in a specific topic, but then how can you apply that in the setting where you are?
I think that obviously your professor was very much minded in that direction. I'm not sure. I would say he's more the exception than the rule. But certainly not. We worked at York with someone called Julie Wilson, who's exceptionally good at working with all sorts of people and was super useful for all of that. And, she's just retired, which is gutting. But it's very hard finding someone else who's equally minded to help in these areas.
Yeah, and of course, people have different mindsets about these things. That's true.
So now we just need to make sure the VIB create that post and give you a career.
Fingers crossed. It's looking good so far, but yeah.
So the other question was, would you move out of flow data as well? Just like the group has, would you move into the imaging side? So working with, Erin now, I think, isn't it? That's leading the core.
Yeah, exactly.
The VIB, or would it go with Evelyn and the transcriptomics data? Would you be looking at, evolving or diversifying to not just flow cytometry, but into the other technologies that are closely associated?
So of course, it's again the never say never, but in first instance, and that's actually a bit what happened after my PhD as well. I see a lot of my peers after they've done their PhD, they're like, go look for another challenge, or they're saying like, okay, this was now enough of this. Let's do something else. I felt like, okay, during my PhD, I've built up all this expertise, and it feels really nice to be able to use that and to build on that. So I feel like that at this point, for sure, I can still contribute useful things to the cytometry field, and that also where maybe it's also part of just being a bit in your comfort zone sometimes, but that having that expertise also makes it easiest to contribute there. We are maybe exploring like very slightly out of that boundary, in the sense like now you have these S8 sorter, which also makes the images. So okay, then you suddenly need a different set of techniques to analyze these images as well. We're looking a bit at the spatial proteomics, maxima, and so on. So like there's also some extension there, but my main focus is still really cytometry. I think like I said, I was introduced to cytometry at the start of my PhD. I kind of fell in love with it. So I think for now I'll stick with cytometry as my main topic. I don't see me like suddenly completely switching to transcriptomics out of nowhere. And I think there are still quite a bit of challenges left in the cytometry field that there keeps being exciting puzzles there as well.
So I'm going to take you back in time now
Okay.
to, I won't say which year, I know, don't go years, but can you remember what the first job is that you ever wanted to be when you were really young? I know you were 6, 7, 8, 9, 10. What was the
I
first
think
job that you thought, oh, I'd like to be that?
when I was very, very little, I think I wanted to be a zookeeper or something like that, where I liked seeing all these animals and so on. I don't think that was a very realistic dream. I don't think we even had any pets at home except maybe a few fish. So I had absolutely zero experience with interacting with animals, but that seemed very, very
Okay.
magical at the time. And then I
That's
think when I was around, yeah, what will it be, 11, 12 years old, I also had a short period where I wanted to become like an Egyptologist or something like that.
yeah.
And then I think looking back on that, that was more the puzzle aspect of it, the unreadable hieroglyphs and wanting to like solve these puzzles. And even though computer science is something very different, I think it's the puzzle solving aspect there that intrigued me about these unknown things from history.
So what got you into computer science?
Yeah, that's actually that love for puzzles, for logic, for reasoning about things, all these kind of things. It's the things I had fun with, like solving math problems. These kind of things were quite fun, and that kind of got me there. And then while I was studying computer science, I noticed also that I really had the interest in machine learning, artificial intelligence, the data analysis aspects of it that was more intriguing to me than just like the computer architecture or these kind of topics that we're having courses on.
And where was your degree, your first degree?
Also in Ghent, so I really stuck to
Oh, you're Ghent.
Yes, exactly. Like I was saying, it's a kind of exception that I'm still so close to where I've been all this time. I've been very lucky that I've had that opportunity.
That must have been UGent and then on to VIB.
Well, no, I'm still, I have the double affiliation, right? So I'm still a UGent.
Ah,
Yeah. Uh,
so,
so
so, so
that's
I,
a
I
long
also,
time at UGent. That's basically been my main institute. I've done a stay abroad of 2 months, and then I had another stay abroad of a few weeks. But like my actual main institute's always been UGent so far.
my undergraduate was also Essex, PhD Essex, first postdoc Essex. So I was
Okay.
so I'm not so dissimilar. So there's
It
nothing wrong.
doesn't happen that often, I have the impression. I think I know, I'm
No.
aware I've been very lucky to have that opportunity in academia where there's a lot of stress often on moving abroad and all these things.
I think it's also lucky if you find yourself in the best research, one of the best research groups. And so
That
actually
for
moving
sure
isn't necessarily moving up because you're losing you if you're in a really good established research group that is highly regarded, actually the opportunities there are really good. And if they're really good supervisors and they let your career flourish, then I think they're really good.
Yeah, that's for sure.
You're a VIB. It is a stunning resource. So, and UGent, and it's a lovely city.
Absolutely. And I think it's also that's then more personal, but I like being relatively close to family and friends. So moving abroad for a longer time, which been more challenging for me, I think.
Mhm. So, thinking about traveling, have you been? You must have been to Cyto.
Yes, I
How
think
many
so.
Cytos have you been to?
Sorry?
How many Cytos have you been to?
How many? So my first site was Fort Lauderdale. I just looked it up. That was apparently in 2014. That was actually the first year of my PhD. And then we were very, very lucky because there was one of these flow cap challenges. And I think it was actually the last one that's been, but there was like a global call of, we have this specific data set and there's a specific question about it. Please submit your results for this. And then we can all learn from each other by doing that. And it was a really great experience. So we knew that the results were going to be discussed at that conference, which at that point was like still for us personally very unknown. But then we were welcomed so warmly. It was really an amazing experience for me. I've been to some, like if you go to a bioinformatics conference, basically everyone's working on transcriptomics and you're just there explaining what cytometry is. Or if you go more to an immunology kind of thing where people know what cytometry is, but then they have no clue what these algorithms are. So you're really explaining that. While at Cyto were the other people who are making algorithms for cytometry. So that was absolutely a great experience. And I think I've been every year since then, except of course the years of the pandemic. And then the first year after, then my son was still too small. So then I also stayed at home. That was Philadelphia. That's the one I missed. But otherwise, I've been every year, and I think that's absolutely the highlight of the year. I really enjoyed Cyto a lot.
Is there still a data analyst meeting within each Cyto? I don't go to that side of it, so I don't know. But is it, if I look at the imaging side, there's always every now and then we'll have a hackathon, and everyone just comes together and talks shop and solves a problem or looks at a few different things. Do we have that for flow cytometry?
So I don't think there's anything like a hackathon up front or anything like that. But it's very nice to see how the program is changing. Where before there was then maybe 1 or 2 sessions about data analysis, software, and so on. And then over the years, that's increasing and increasing. And now you can like completely fill your Cyto program just with data analysis related sessions. And that's nice to see also how the impact on the community is changing with all the high parameter data being generated right now. People have more and more of a need for all these tools. So even if you now go to a more whatever other topic focused session, there will probably be some discussion about the algorithms used to tackle these issues, which you would not have had a few 10 years ago. Yeah.
I'm too late for this year, obviously because Denver's well being put into place. But I know in the imaging and the flow world in the UK, we have a flow cytometry facilities meeting.
Yeah.
So all the flow staff, we got the European virtual flow meeting where all the flow core staff meet. I don't know if you attended that.
My colleague participated. Sarah was there.
And then like microscopy similar, but actually wouldn't it be great to have a flow data science similar type of meeting where everyone just talks about funding, how job recognition, career paths. Oh, actually in the UK we have something called the Technology Specialist Network, and they look after those technology specialists, which includes data scientists, to make sure the career paths are now being properly developed. Because look, you're too young to know this, but 20 years ago these jobs didn't really exist, and now they're very much at the forefront because they're pivotal now to underpin the science. They're absolutely vital, but not necessarily the careers are being properly established. And that's what I think we spoke about a bit earlier. And you're an obvious person who needs to make sure of that. But even if you succeed, you then need to share that to make sure others can succeed within their institutes.
Yeah, sure. I think it's something that's slowly becoming relevant. I think if you, like 10 years ago, there were not enough people around doing computational cytometry to really warrant that. And then it's nice to be embedded in the larger conference. It's still nice to be embedded in the larger conference. But I think there's more and more room for that, where more and more people are actually working on this topic and making great changes there. Yeah.
It'd make a great pre-Congress meeting, though. So if you think about it, talk about day zero with the pre-Congress on Saturday, Sunday, that would be an ideal session to bring everyone together to talk shop.
Something to think about, yes. I think what it's different, of course, in AIM, but we're also last year, we for the first time organized this computational cytometry summer school, where we had a week that was because Cyto was in Europe, we kind of had that the week before. I also learned never organize something you're heavily involved with like the full week before Cyto. That's just too much for all the things you also want to be preparing for Cyto. So that was a lot at the same time. But it was a lot of fun, and we're doing it again this year, not the week before Cyto, because Cyto is in US anyway, so that wouldn't make sense. And yeah, that's also fun to see how many people are interested in learning these things, and how many people are picking up all these tools and willing to dare programming in R to be able to get things running. I think that's really cool to see. So that's also a very fun environment to be in. Yeah.
I have another question. So actually, from my side, I had a PhD student called Laura Wiggins. With Will Brackenbury and Julie Wilson, who I mentioned earlier, who three different type of. I'm the imaging cytometry person. I had someone with a biological question, that's Will. I had Julie, who had the data science.
Yeah.
And Laura was a master with computer science. Never done any biology, just like yourself as well.
Yeah.
But one of the first things we did was get her in the lab and generate her own data as well. So actually, have you been in the lab? Have you generated your own flow cytometry data?
Have you generated your own data is maybe a bit too much of a stretch, but I did follow along, like a day in the lab, following along with someone, being allowed to do some of the non-crucial steps that can't really go wrong, even if you're not fully sure of what you're doing, kind of thing. And I think that's been very informative. I really appreciated that a lot. I've had that opportunity a few times by now. And every time you get more insights about how the data is generated, which does have an impact on your downstream analysis. So I think that's absolutely crucial to understand what you're actually doing, right? It's not just numbers, they're coming from somewhere. And I think on the one hand, to be aware, for example, which kind of quality control makes sense, which kind of quality control is needed. You get informed a lot by actually following along the experiment measurements. But on the other hand, one additional advantage of it is just to be able to talk to the researchers generating the data, to understanding their point of view, and also to be a bit more understanding sometimes, maybe like we can complain of, oh, why is it not perfectly standardized? Why is not everything typed exactly the same? And that till the point where you're following along in the lab and you're understanding like, oh, wait, this is not so straightforward than it is for me afterwards when I'm seeing the data. So I will keep encouraging everyone to try to standardize things and so on. But it also helps a lot with understanding that what happens in the lab is sometimes unpredictable, all these things. And seeing that firsthand really makes a difference in how you can treat that afterwards. Yeah, absolutely.
But you kind of answer my next question, because that's exactly why we got Laura to get her own data, so she can understand the provenance, the limitations. Because I can imagine, and you said sometimes it means you won't ask the users to do things that aren't really possible. And you understand why the data is frustrating, because not everything in the data is as you want it to be, just because biology doesn't allow it. I think you're absolutely right. So those days' experience are what have given you that insight to understand that biology is pretty non-reproducible to a large extent. Samples are very limiting. You can't get lots and lots. Sometimes it's very, very precious, and you don't statistically challenging.
Yeah, exactly. And I think being aware of that, and the best way to become aware of it is seeing it firsthand, really helps a lot in how you tackle these things. I
What would
fully
you
agree.
say has been the hardest time in your career to date? You must be one of the youngest people I've ever done a podcast with, but there's always challenging times, especially early in a career. So when would you say the most challenging time has been?
That's a good question. I think maybe one of the challenging time for me is like the switch from PhD to postdoc, maybe, or like the short period after that. Because maybe also because I stayed in the same research group, right? I kind of kept doing the same thing, but I was more and more guiding people. But that felt like not really doing my work, because it was not like, I still have like some own research projects, and then more and more also following along with other people and thinking along with them, which I enjoyed a lot, which is also why I'm still doing it. But which had sometimes the thing like, oh, but what did I actually do this week? My whole week flew past and I did not do anything, and that created a certain level of stress. And getting to reframe that in my head of, okay, but actually guiding these people is the main function I'm doing now, and it's okay that I don't get to programming in R every day of the week. So that's period where I was like slowly growing from one function to the other came with some moments of stress for sure.
Okay.
I
And I've got to ask, so those, I know most people are listening to this, but I can see Sophie's background. Is that a plate of peas as your background?
I don't know. I'm in this meeting room here. I think it's some kind of moss.
It looks like a plate of mushy peas, and it's just a bit of wall art in the background.
it's a bit of wall art that should represent moss. I think I can put, like, there's another one over there.
Okay. I've never seen that when I've been there.
It's
I have
just
eaten
how
one.
our meeting rooms here in the institute are decorated.
So a complete digression. So you say you have a daughter or a son?
A son. He's about 3 years old now. Yeah.
And how have you found managing the work-life balance?
I think it's actually relatively okay, in the sense that I was already in the habit of not working too many hours from home. So I stay quite strictly to my working hours for my job. Of course, there's always some exceptions if there's a specific deadline or whatever, but like, I think compared to some colleagues, and I think at some moments this also causes you stress, like, should I be doing more hours, this or that? But then in the end, you notice like, no, this is actually what is expected of me, and that is fine. So in that sense, it's been quite doable. However, since this September, he started going to what do you call it in English? Kindergarten, then I guess, but like
Yeah.
school. And then I don't know how this is abroad, but in Belgium, so Wednesdays they only go half a day. And then they could go to a kind of daycare solution in the afternoon, but I decided to go work 4/5. So I'm home on Wednesdays to take care of him. And so that switch from working full-time to 4/5 has been a bit challenging, because actually, like, okay, I have quite a bit of control of what I'm doing with my days, so I just need to learn to estimate better that you can't do everything you were doing in 5/5 and 4/5. And so that also comes with its challenges sometimes, like, where did this week go? It's already passed, because you're missing a whole day every week, right? But otherwise, next to that, I think it's been quite okay. Yeah, and I also have the feeling that we're in a very how to say it, accepting environment. Like, people are understanding if you need to leave early a day, or you need to suddenly miss a day because the kid is sick and you need to be at home, or things like that. Like, people are flexible about these things, and they're understanding about these things. So that's again being a good place to be at here in the institute, for sure.
So I asked about the most challenging times. You talked about more challenges there. What about if you could go back and relive any one year, which year would you go and relive?
Oh, a specific year to
Well,
relive?
it doesn't have to be the year itself.
No,
What
but
sort of
like
thing?
a
First year of a
time period.
PhD, the first year as your postdoc, The year you've just had, the year you went to Cyto. What was the most fun year that you think, wow, that was a blast?
That's a difficult question. I think every year comes with some ups and some downs, and I'm for sure also looking forward to the future. So I'm not one of these persons of like the best is in the past or something like the best is yet to come, hopefully. But I think like the content-wise, probably the start of my PhD would be the moment I would refer to, where you're just starting to explore all these things. You have quite a bit of freedom in all the things you're doing. You don't have too many commitments yet that you need to uphold. So yeah, if I have to pick something, I would say that, but it's certainly not that I'm here like, oh, I wish I could go back there. No, I'm very happy to just keep moving forward.
So I'm going to ask some quick fire questions.
Okay.
Okay. So are you an early bird or a night owl?
Slowly becoming more and more early bird. Would have been different a few years ago.
PC or Mac?
Oh, PC. Yeah.
Oh, okay. McDonald's or Burger King?
I've never been to a Burger King. McDonald's sometimes, but also not very often. So I'm okay with neither. Like, don't need that either.
You have a 3-year-old that will go up in numbers.
Sure.
What is your favorite takeaway then? What's your favorite fast food? Your favorite takeaway?
When I was younger, I would have said Chinese, but now I'm eating vegetarian, and at least here in Belgium, you don't have that many options there. So now I'll probably just go for fries.
Good. Oh, it is Belgium, I suppose. You can...
Yeah. Look, there's some traditions to uphold, right?
One of the two. Tea or coffee?
Tea, for sure.
Chocolate or cheese?
Would miss both a lot if they would not be there. Let's say cheese wins.
Okay. Beer or wine?
Neither. I don't drink any alcohol.
Ah, and you're in the land of trapeze beers. Oh, of any country, it's the one country you have to have beer. Fair enough.
Makes up for it with the chocolate.
What is your favorite food?
Favorite food? It's also a difficult question. I think I like variety, like not having to eat the same thing every day. I don't have a very strongly spoken favorite there.
Is there any food you really dislike, other than obviously meat? But is there any food that the taste is just... Imagine you went to Cyto and you went for the evening meal, and that was on the menu to go, oh no, how am I going to eat that?
I'm trying to come up with something. No, I think the main disappointment was be like, oh, there's no vegetarian option or things like that. So, yeah.
Okay. Do you like to eat in or eat out?
I enjoy eating out. Yeah, we don't do it that often, but yeah, then the times it happens, it's nice.
Would you rather cook or clean?
Preferably neither, but if I have to choose, I guess cleaning. I'm not the best cook.
So is your partner the cook at the house?
Yeah.
And what's their signature dish?
Oh, I don't know. I think we like...
What's the favorite dish that they cook for you?
Also a good question. I think it's also just the variety. We're also like, it's also not that his big ambition is cooking or his big passion is cooking. So we're kind of just making do with easy dishes that are ready quickly. And that's why we then enjoy eating out once in a while, right? When neither of us have to cook.
Because my next question to that was going to be, what does he cook that you least like?
Yeah, also don't have a very specific answer to that. I think it's I'm sorry, I'm very bad at saying names of
I'm just
dishes.
trying to get you into trouble because he'll probably watch this back and he'll go, why you never told me you didn't like that? And then you forget it again. Oh, there was your
I think it's
never have it again.
Sorry?
There was your opportunity to say something, and you never have to eat that again.
Yeah. No, I don't think there's anything like that. That's still on the menu list right now.
TV or book?
Books for sure. Yeah.
What are you reading at the moment?
I think the last thing I read was a very silly rom-com genre book, but then before that, some nice science fiction. So a balance of those, let's say, in general. And once in a while, a non-fiction book, but that's only once in a while, mainly fiction. Yeah.
Okay, and what's your favorite film? Favorite movie?
Oof, that's also a very difficult question. Maybe my answer can be more that it's really been quite a long time since I watched any movies. Like the question from like television or books was a very easy question. I really don't watch a lot of television.
But you must come on. It was before Edinburgh. It was Montreal. Surely you watched movies on the plane.
Not that I remember right now, but probably something, but nothing that's stuck enough to remember right now. And the probability, like actually on a plane that I just sleep the whole time is also quite probable.
Oh, wow, you're a good sleeper on long haul flights. That's
I think I don't know which one it was. It was coming back from the States, but like it was not Montreal, it was already longer ago. But where I just like I was not fully awake yet anymore, I think, when we still had to lift off and then we landed and we were there and I kind of missed
it
the
was in the
whole
East
flight.
Coast, one of the East Coast meetings then. So it's
Probably, yes.
awesome. Okay. Star Wars or Star Trek?
Um I think I've seen more Star Wars than Star Trek, but I'm not strongly big fan of either. And I think probably from the reasoning point of view, I would prefer Star Trek over Star Wars.
Okay. Uh, music. What's your favorite music style?
Oh, actually, I'm listening more and more to classical music lately, which I didn't really do as a teenager or something, but like lately that's bringing back some calm and some nice memories from Sunday mornings as a kid and so on. While then you think like, oh, why can't we listen to my more fun music rather than the boring grown-up music? But I'm really getting back to that more and more now. Yeah.
Favorite color?
Let's say green.
Fitzy green or GFP green?
What?
Fitzy green or GFP?
Oh, no, that's like, therefore I'm too far away from the instrument to to have an answer for that.
Okay, so moving on. Who would you say has been your biggest inspiration in your career so far?
That's also a difficult question, right? I think just in general, all the computational cytometrists, so all the people at Cyto that are really showing what change algorithms can make. I'm thinking of Brian Brinkman, Nima, all these people that there's many of them have been a big inspiration for sure of believing that this is a field like that it's okay to focus on this one field of cytometry and trying to see what difference we can make there. And then I think otherwise, and it's maybe a bit more vague, but all the people who show me that the work can have an impact, so all the people I'm collaborating with. And then I'm actually also thinking in particular of Gert, so the head of our core, who's always there and saying, yes, we need these kind of things. That's for sure also been a big inspiration.
But Gert's a big name in the flow world as well. So I guess, and you've been there a fair while, so and he's developed his name very strongly since he's been at the VIB. So
I think like my first Cyto was also his first Cyto, as far as I'm aware.
So I can imagine anyone new going into VIB now might find it quite intimidating, just because he's such a big name. Whereas actually he's lovely. He's really cool.
That's what I would say as well. Yeah. No need to be intimidated. I think we're all very happy for new people to learn about all these things, to start exploring these things, to have fun with it, to do amazing discoveries with cytometry. Absolutely.
So I asked inspirations. Is there any What's your biggest motivation? What motivates you? Who motivates you? What motivates you?
I think my motivation, if I like look back on things, is mainly having other people being happy with things. So seeing people being able to use the tools that I developed, like seeing Flowsum popping up on work of people I've never heard of, that's amazing, knowing that you contributed something. And then also just in the collaborations, I think if I have to do work on my own, just for myself, I'm a whole lot less motivated than if it's a group effort, or I know that someone else is involved who also cares about the project. That makes a big difference for me.
If this was an interview to work in the core facility, you'd be doing very well with that answer. It's very much about.
That's also why I was thinking, like, this seems like a good option if that could become possible, right? That's the kind of thing I'm getting excited about.
Do you have any pet hates? What annoys you? What little things annoy you? Not necessarily in work, in life in general. What are your pet hates? What things annoy you?
That's a good question. I think when people are too strict or like don't have enough empathy for someone else, where they're like getting very like, oh, why doesn't this person do? Yeah, but you need to hear their part of the story as well, right? And there's probably a good reason, and things like that, or where there's like, yeah, I don't know, people being very like, oh, but this is the way it should be done, and can be the only way, and then yeah, but in some situations there is a good reason for an exception or these kind of things. So being too strict in that sense, I think, would be the main thing that comes to mind.
I quite like that, because that's almost having an intolerance to intolerance.
Yeah, but that's I think that's fair. I think that's a good thing. That's the one thing to be intolerant about, is too much intolerance. I think that's a good point of view.
I fully agree. Yeah, I find intolerance.
That's very reassuring to hear.
Yeah. So outside of work, what hobbies do you do?
Yeah, so we already mentioned books. I like reading quite a bit. Since we like have our own house, I also quite like gardening. My parents had a very nice garden with lots of flowers and so on, and I was never really actively involved in that as a kid. I was mainly like somewhere inside reading a book. But then once I moved on my own, it was like, yeah, if I don't do anything in the garden, there will not happen anything in the garden. And then I really started enjoying that as well. So that was also very nice to like more reconnect with my mom in that sense. Yeah, and I think that would be the main two things right now. Yeah, and of course, enjoying time with family, with my cats. Yeah.
You said you don't have pets, so your garden's going to be easier to maintain, I guess.
No, so I have a cat. I have a cat now. Yeah, yeah.
Which doesn't
only got it like 5 years ago.
Okay. So what sort of garden is it? Is it sort of plants? Is it shrubs? Is it trees? Are you growing your own
trees. So
food?
where we're currently living, it's a quite small garden, but it's fully planted with plants. So there's no lawn. It's all mainly smaller plants. A few edible things, but also not that like. I do that for the fun of it, that it's fun to make to grow something that you can eat, but not for like our daily consumption or anything. That's more as a hobby project than as an actual goal of not having to go to the supermarket kind of thing. But so yeah, lots of flowers, things like that. We will move still quite a while to go, but then we'll have a bigger garden, so then we'll be able to combine some lawn with hopefully still lots of plants as well. It's exciting. So and then maybe even some room for trees. We'll see. Some smaller trees should for sure be possible then as well.
So I like your future. You're already thinking about house, what that's going to be like. The garden is obviously a big prerequisite. If I was to ask you, we're coming up to time. If I was to ask you, what would you see as the biggest challenge in the world of data science and flow cytometry? Or what would you like to see be solved? What would you like to be easier? Any of those?
in cytometry, right? And I think actually maybe this is a very nice moment to give a shout out to the SoulCap initiative, where we're trying to standardize the annotation of cytometry data. And I really very strongly believe in that. So right now, everyone has their own gating strategy. And when one person is talking about a certain cell type, they might mean something else than someone else talking with a population with the same name. And then especially with all the clustering and so on, it's also like, okay, you have a cluster result, but which cells are these now? That's still a whole other question. So I think that's one of the big next questions to be solved. And if anyone would be interested to working on this with the SoulCap initiative, we're really making a huge effort at this, and we would be very happy for anyone to be involved in that. So then for sure, reach out about it. That would be great.
Are you on any of the ISAC committees?
I'm on the data committee. It has not been very active lately. I hope that that will pick up again at some point. I was also on a task force at some point about CytoU when all the webinars were going to be labeled and so on. So I helped a bit with the data science related ones there. Yeah, but so mainly the data committee. And then now there's also this task force being started more about the data analysis education. So more fitting in the flow content committee there. I'm also excited about that, bringing more awareness about the algorithms to all the people using them who are now maybe just clicking a button without really knowing what they're doing. So bringing that message across is also exciting.
There's quite a lot of potential there.
Yeah, exactly. So that's
if they're
what
not
we're
doing those things at the moment.
That's what we're trying to make happen. Exactly.
Take it, grab it, and go for it.
Yeah, exactly. That's why the subcommittee or task force or whatever it's called is being started right now. Yeah.
And you know what, task forces I think could be better than committees, because task forces are liberated. They don't have the bureaucracy of a committee. They can just get on and do an action and actually deliver things much better.
I think there's value in both, right? So you need to have both. But it's true. Like I think with the task forces are more actionable, right? You have a concrete plan and you go for it, and then it gets done. While the committees are a bit more on long-term vision and these things. It's two different things that both have a lot of value, I think, for the community.
So Sophie, I think I've come to the time point for the podcast. Thank you so much for joining me today. Everyone who's listened, watched, thank you for go back and look at the other Flow Stars. Sophie mentioned Ryan Brinkman. Go listen to his podcast. He was utterly brilliant. You've heard about Isaac and Cyto, and we've got Virginia Litwin as one of the cover guests of this season that you're listening to now. But you can go back and listen to the previous presidents, such as Jessica Houston, Johnny Moore, Paul Smith, Paul Robinson, Wallace, the lot of them. But Sophie, today, God, it's exciting having you in the world, isn't it, of flow cytometry? I think we're so fortunate to have yourself and others like you really coming to the fore and putting your expertise in our world for the data science and being passionate about it. And I really hope you get that job in the core, because I think you'll be brilliant at it. Sophie, thank you very much.
Thanks so much for having me, especially in this long list of names. It feels like a bit intimidating to be among all those great names, but it was a very nice opportunity. Thanks so much.
It's a pleasure. It would have been great to have them at this stage of their career. So this is why this is so important. Sophie, thank you.
Thanks so much.